Welcome back, everybody, to the deep dive. This week, we're going to do another deep dive into some really interesting longevity papers
Yeah.
that, just came out and are really kind of pushing the field in new directions. We've got discoveries from gut health and altitude to even gene editing and how DNA changes as we age.
Yeah.
Are you ready for this?
Absolutely. I think there are some really unprecedented findings here.
Okay, well, let's jump right in. One that really caught my eye was this paper that was published January 13th in Advanced Science Researchers from Chongqing University in China, and they were looking at the connection between gut health and ovarian aging.
And the really remarkable thing about this study is that they were able to show that transplanting a specific gut bacteria, Bacteroides fragilis, actually reversed some of the signs of reproductive aging in mice.
Reverse, that's a big deal, huh?
So they found that Bacteroides fragilis delivers these little packages called extracellular vesicles that travel from the gut to the ovaries, and they carry a molecule called miR-1246 that seems to directly reduce oxidative stress in the ovaries.
So it's like a targeted delivery system for anti-aging compounds.
Exactly. And, it raises this really fascinating question. You know, could tweaking our gut microbiome be a key to extending reproductive lifespan in humans? I mean, this research is really opening up a whole new avenue for exploration.
Yeah, it's really interesting to think about what the implications might be. If we could somehow influence, reproductive lifespan through something as simple as, diet or probiotics. Okay, well, let's move on to our next paper. This one takes us to high altitude. This was published on January 9th in Geroscience researchers at the University of Copenhagen. And they were using deep learning models to analyze how living at different altitudes affects aging. And they looked at this in Ethiopia.
Yeah, and the interesting thing about this is they found these kind of paradoxical results. They found that higher altitudes were linked to both a decrease in disease burden, but also a faster rate of facial aging.
So it's like a trade-off. You might live longer, but you're going to look older faster.
It seems that way. The researchers think that maybe the lower oxygen levels at higher altitudes might be protective against some age-related diseases. But then the increased UV exposure you get at those higher altitudes accelerates visible signs of aging.
Huh. That's really fascinating. Just to think about how the environment can have these very complex effects on lifespan and how we age. Okay, let's stick with neuroscience for a minute, but shift our focus to the brain. Researchers from Tehran University of Medical Sciences published a paper also on January 9th, and this is looking at the impact of creatine supplements on cognitive decline in aging mice.
Yeah, creatine, it's most often associated with athletic performance, but this research suggests it could actually play a role in brain health as well. And so what they found was that supplementing the diet with creatine boosted the activity of a key brain enzyme called CKBB, which is really important for brain energy metabolism.
So are they suggesting that creatine could actually help keep our brain sharp as we age?
Well, they found in the study that the mice who received creatine had improvements in learning and memory, and they even had protection against damage to synapses, the connections between brain cells. So it's a promising avenue for further research.
Yeah, it's definitely changes the way I think about creatine. It's not just, a muscle building supplement.
Yeah.
Okay, this next paper feels like something straight out of science fiction. This was published January 15th in Nature Medicine, and researchers from the Broad Institute at MIT and Harvard actually extended lifespan in a mouse model of prion disease using in vivo base editing.
Yeah, this is really groundbreaking work. Prion diseases, they're fatal neurodegenerative diseases that are caused by these misfolded proteins. And what these researchers were able to do was actually use this cutting-edge gene editing technique to target and correct those misfolded proteins directly.
So they're basically fixing the root cause of the disease at the genetic level.
Exactly. And the fact that they were able to do this in living animals using in vivo base editing is really a huge leap forward. It opens up the possibility of using similar approaches to treat other neurodegenerative diseases like Alzheimer's and Parkinson's.
Yeah, it's almost hard to wrap your head around the potential implications of this.
Yeah.
If we can target and repair the genetic errors that cause these diseases, it could really revolutionize how we approach treatment.
Absolutely. It's a game changer.
It is a game changer. Okay, this last paper in this section takes a fascinating look at the connection between DNA mutations and epigenetic age. This was published on January 14th in Nature Aging by researchers at UC San Diego, and they were looking at how those random changes in our DNA that we accumulate, called somatic mutations, how they relate to our epigenetic age.
Right, and just to remind everybody, epigenetic age is essentially a measure of biological age based on changes to the way our genes are expressed rather than chronological age.
Oh, what?
And so what this study found was that these random DNA mutations, they might actually be a driving force behind these epigenetic changes we see as we get older.
Hmm. So it's not just that our DNA is accumulating damage over time, it's that the damage itself is actually influencing how our genes are expressed and therefore how we age.
That's what this research suggests, and they actually developed a method for predicting age based on these mutations, and it matched up surprisingly well with the existing epigenetic clocks.
Huh. That's really interesting just to think about the role of these random mutations. Maybe they're not just passive byproducts of aging, but active players in the process.
Right, it's a really thought-provoking idea.
It is. Okay, well,
Yeah, it really challenges our understanding of how aging works at the molecular level.
it does. It adds another layer of complexity to the picture.
Hmm.
Speaking of complexity, let's talk about epigenetic clocks.
Hmm.
There was a fascinating paper that came out January 9th in Aging Cell researchers at Penn State University, and they were comparing epigenetic age predictions across different tissues, so blood, skin, muscle, fat.
Yeah, and what they found was really intriguing. The accuracy of those epigenetic age predictions varied depending on the tissue type. So some clocks were better at predicting age in blood, while others were more accurate for skin, for instance.
So does that mean that different parts of our bodies might be aging at different rates?
That's one interpretation. It really kind of challenges this idea of a single unified biological age. It raises the question of, should we be looking at aging on a more granular level? Should we be thinking about, the age of individual organs or systems rather than the body as a whole?
Yeah, that's a really interesting concept. What does this mean for how we, measure and track aging? I mean, do we need to develop, more specialized epigenetic clocks that are tailored to specific tissues?
I think this research certainly points in that direction. It suggests that a one-size-fits-all approach to measuring biological age, might not be the most accurate or informative.
Right. Yeah, it seems like we're only just scratching the surface of understanding how aging works, at the cellular level. Okay, let's move on to a paper that dives into the brain, specifically how our brains eliminate synapses as we age. This was published on January 21st in Cell Death and Differentiation by researchers at University College London.
So, synapses are those connections between brain cells that allow them to communicate with each other. And we know that we naturally lose some of those connections as we age. And it was always thought that this was just a detrimental process, part of the decline of aging. But this research suggests a little bit more nuance than that.
Okay, so it's not just that we're losing brain cells, but also the connections between them.
Exactly. And this study really focused on the role of glial cells, which are the support cells in the brain. And they found a specific gene in these glial cells called Croquemort or Croquemort that seems to play a really crucial role in this process of synapse elimination.
So what happens when this gene isn't functioning properly?
So they found that when Croq is absent in glial cells, the brain actually retains these excess synapses even into adulthood. And you might think that, having more synapses would be a good thing, but it actually led to problems like seizures in the mice they studied.
So it's a case of too much of a good thing.
It seems that way. There's this delicate balance that needs to be maintained. And interestingly, they also found that the loss of Croq in these glial cells actually prevents age-related synapse loss.
So could this be a potential target for, preserving brain function as we age?
It's certainly a possibility that warrants further investigation. If we can understand how to precisely modulate this process, it could have really significant implications for treating age-related cognitive decline.
Yeah, that's really exciting. It seems like every week there's new research that offers, hope for extending not just our lifespan, but our health span as well.
Yeah.
Okay, this next paper caught my eye because it explores the connection between our muscles and our brains, specifically in the context of Alzheimer's disease. This was published on January 10th in Alzheimer's and Dementia by researchers at the USC Leonard Davis School of Gerontology.
Yeah, this is a fascinating area of research that's really gaining a lot of traction, this idea of a muscle-brain axis. So essentially what that refers to is the communication pathways between our muscles and our brains and how they influence each other.
So it's not just that exercise is good for our brains, but that our muscles are actually sending signals that can directly impact brain health.
Exactly. And this study looked at a mouse model of Alzheimer's disease, and they found that activating this muscle-brain axis actually reduced amyloid plaque accumulation in the brain.
Wow, it's huge. Amyloid plaques are one of the hallmarks of Alzheimer's disease, right?
Yes, they are, and they're thought to play a key role in the development and progression of the disease. And what's even more impressive is that activating this muscle-brain axis also improved cognitive function and enhanced the integrity of synapses in these mice.
So how do they activate this muscle-brain axis?
So they overexpressed a gene called TFEB in the muscles of these mice. And TFEB is what's called a master regulator of autophagy, which is basically the cell's way of cleaning up and recycling damaged components.
So by boosting autophagy in the muscles, they were able to protect the brain from the damaging effects of Alzheimer's disease. That's incredible. Does this mean that, exercise, which we know boosts autophagy, could be a potential therapeutic strategy for Alzheimer's?
It's a very promising avenue for further research. This study really provides compelling evidence that targeting this muscle-brain axis could be a powerful new approach to treating neurodegenerative diseases.
Yeah, it's amazing to think that something as simple as exercise could have such a profound impact on brain health. Okay, this next one takes us back to the brain, but this time we're looking at how protein aggregation changes with age. This paper was published January 10th in bioRxiv, the preprint server for biology by researchers at the University of Tokyo.
Right. So protein aggregation is a hallmark of many neurodegenerative diseases, but it also occurs to some extent in the brains of healthy individuals as we age. And this study was really focused on trying to identify, proteins that become insoluble with age. And they found one called HAPLN2 that specifically aggregates in the cerebellum, the part of the brain responsible for coordination and movement.
So what does this protein do, and why does it aggregate with age?
So HPLN2 is an extracellular matrix protein, meaning it's found outside of cells and spaces between them. And it seems that the brain's ability to clear away these protein aggregates declines with age, and so they start to build up.
And what are the consequences of this buildup?
So they found that these aggregated HAPLN2 proteins can trigger inflammatory responses in glial cells, potentially contributing to neurodegeneration.
So this protein aggregation could be part of the reason why our motor skills and coordination decline as we age?
It's a plausible explanation. This research really sheds light on the molecular mechanisms underlying cerebellar aging and highlights the importance of maintaining a healthy brain environment to prevent this protein aggregation.
Yeah, I'm sensing a theme here. A lot of these papers are pointing to the importance of these cellular cleanup processes, like autophagy in maintaining health and potentially slowing down aging.
You're absolutely right. It's becoming increasingly clear that our cells' ability to efficiently remove, waste and damaged components is playing a really crucial role in how we age. And finding ways to boost those cleanup processes, could be key to extending our health span.
Yeah, it's a whole new way of thinking about anti-aging interventions, focusing on enhancing our body's natural repair and maintenance mechanisms rather than just trying to, fight off specific diseases.
Exactly. And speaking of enhancing those natural mechanisms, let's move on to a paper that looks at the link between gut bacteria and cellular senescence. This one was published January 9th in Nature Aging.
Cellular senescence, that's when cells stop dividing and enter this kind of, retirement state, right? I think we talked about that in a previous episode.
Yes, and it is a natural process, but these senescent cells, they can accumulate and start to cause problems as we age. They can release inflammatory molecules that damage surrounding tissues and contribute to all sorts of age-related diseases.
So what did this study find about the connection between gut bacteria and cellular senescence?
So researchers from the National Clinical Research Center for Aging and Medicine in China found that a specific gut bacteria metabolite called phenylacetylglutamine, or pegrilin, actually accelerates cellular senescence.
So a byproduct of our gut bacteria could be contributing to the aging process.
That's what this research suggests. They found that levels of pegil increase with age, and that blocking this metabolite can actually prevent or delay cellular senescence in cell cultures.
So could this open up new avenues for anti-aging interventions that target the gut microbiome?
Absolutely. This study provides further evidence that what we eat and the trillions of bacteria that reside in our gut can have a really profound impact on our health and longevity, and understanding these connections could lead to all sorts of novel strategies for slowing down the aging process.
This is also fascinating, from gut bacteria to protein aggregation. It's amazing how interconnected these different aspects of aging seem to be.
Yeah, it really highlights the complexity of aging and the need to kind of look at it from, this systems biology perspective. Everything is connected.
Yeah, it's like our bodies are these intricate ecosystems, and we're only just beginning to understand, how all the different parts interact and influence each other.
That's a great way to put it. And speaking of ecosystems, let's shift gears and talk about a paper that explores, a really innovative approach to rejuvenating bone regeneration in older individuals. This was published on January 13th in Advanced Healthcare Materials by researchers at Chongqing Medical University in China.
Bone regeneration. Now, it's not something we've talked about much on the show. What's the story here?
So as we age, our bones naturally become less efficient at repairing themselves, which can lead to, an increased risk of fractures and other bone-related problems. And so these researchers developed this really interesting hydrogel that can actually restore the bioelectric signaling in bone tissue, which promotes blood vessel growth and stem cell differentiation.
So they're essentially using this hydrogel to kind of jumpstart the bone healing process.
Exactly. And they found that it significantly enhanced bone regeneration in an animal model of aging, almost like turning back the clock on bone healing.
Wow, that's amazing. That could be a game changer for older adults who are at risk of fractures and other, bone-related complications. Okay, this next paper caught my eye because it involves something we all enjoy, mushrooms. This was published on January 11th in NPJ, aging researchers from Kumamoto University in Japan, and they were studying the effects of long-term consumption of tomogi-take mushrooms.
So tomogi-take mushrooms are known to be a good source of ergocyanine, which is a potent antioxidant.
And antioxidants are those molecules that help protect our cells from damage, right?
Precisely. They help neutralize these harmful free radicals that can accumulate in our bodies and contribute to aging and disease.
So what did they find about these mushrooms?
Well, they fed these tomogi-take mushrooms to mice over an extended period of time, and it actually improved their heart function, enhanced their exercise capacity, and even increased their lifespan.
Wow, that's impressive. So are they suggesting that adding these mushrooms to our diet could help us live longer, healthier lives?
This study provides further evidence that dietary interventions can have a really significant impact on health and longevity, and that, certain compounds found in foods like ergothioneine may play a protective role.
Yeah, it's a great reminder that, what we eat can truly be medicine.
Absolutely. And with that, we've come to the end of another deep dive into the fascinating world of longevity research.
Wow, we've covered so much ground today, from gut bacteria and gene editing to bioelectric signaling and the power of mushrooms. It's incredible to see the progress being made in this field.
Yeah, it really feels like we're on the cusp of a major breakthrough in our understanding of aging and how to extend not just our lifespan, but our health span as well.
Absolutely. It's an exciting time to be alive and an even more exciting time to be following this research. Who knows what amazing discoveries await us just around the corner. Until next time, keep diving deep.